DNA replication. Detailed pointwise explanation. 1. Definition and purpose. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. Occurs during S phase of interphase in the cell cycle. Essential for genetic continuity. Ensures each daughter cell receives identical DNA. 2. Site of replication. In eukaryotes, nucleus. In prokaryotes, cytoplasm. Replication begins at specific DNA sequences called origins of replication, ORI sites. 3. Nature of replication. DNA replication is semi-conservative. Each daughter DNA molecule contains one parental strand and one newly synthesized strand. DNA replication occurs in three main steps. Step 1. Initiation. The process begins at specific sites called origins of replication. The enzyme helicase unwinds the DNA double helix, forming a replication fork, while other proteins stabilize the strands and prepare them for copying. Step 2. Elongation. DNA polymerase adds new nucleotides in the 5' direction, using the original strands as templates. The leading strand is synthesized continuously, while the lagging strand is formed in short segments called Okazaki fragments. Step 3. Termination. Once replication is complete, RNA primers are removed, gaps are filled with DNA, and the fragments are joined by DNA ligase to form two identical DNA molecules. These three steps ensure that genetic information is accurately copied and passed on during cell division. DNA replication, three main steps. 1. Initiation. Where it begins. At specific sites called origins of replication, ORI. Key processes. Helicase unwinds the double helix by breaking hydrogen bonds. Topoisomerase/gyrase relieves supercoiling. Single-stranded binding proteins, CBSEs, stabilize separated strands. Primase lays down RNA primers to start synthesis. 2. Elongation. Main synthesis stage where DNA strands are copied. DNA polymerase adds complementary nucleotides in 5' direction, using leading strand synthesized continuously, lagging strand synthesized discontinuously as Okazaki fragments. Multiple enzymes work together, helicase, primase, DNA polymerases, etc. 3. Termination. RNA primers are removed by DNA polymerase I in prokaryotes, RNase H and Fin1 in eukaryotes. DNA polymerase fills the gaps with DNA. DNA ligase joins the Okazaki fragments by sealing the nicks in the DNA backbone. Replication ends with two identical DNA molecules. Major steps in DNA replication. Step 1. Initiation. Helicase enzyme unwinds the double helix by breaking hydrogen bonds between base pairs. This creates a replication fork, Y-shaped structure. Topoisomerase, or gyrase in prokaryotes, relieves supercoiling ahead of the fork. Single-stranded binding proteins, SSBs, bind to the separated strands to prevent re-annealing. Step 1. Initiation. Detailed explanation. 1. Helicase action. Unwinding the double helix. Helicase is the first key enzyme to act during DNA replication. It binds at the origin of replication, ORI site, and moves along the DNA, using energy from ATP hydrolysis. Its function is to break the weak hydrogen bonds between complementary base pairs. A equals T and G is identical to C, thus unzipping the double-stranded DNA. This exposes the two single DNA strands, which act as templates for new DNA synthesis. 2. Formation of replication fork. As helicase unwinds the DNA, it creates a Y-shaped structure called the replication fork. The replication fork has two single-stranded DNA templates. One strand moving toward the fork, leading strand. One strand moving away from the fork, lagging strand. Replication forks form on both sides of the origin, making replication bidirectional. 3. topoisomerase, gyrase in prokaryotes, preventing supercoiling. As the DNA unwinds, it creates tension, torsional strain, in the remaining coiled DNA ahead of the replication fork. This tension leads to positive supercoiling, which can block the replication process. Topoisomerase enzymes solve this. They cut one or both strands of DNA, relax the coil by allowing controlled rotation, then rejoin the strands. In prokaryotes, the specific enzyme is called DNA gyrase, a type 2 topoisomerase. 4. Single-stranded binding proteins, CBSEs. Once the DNA strands are separated, they tend to reanneal, rejoin, or form secondary structures like hairpins. SSB proteins bind to exposed single-stranded DNA to stabilize the strands, prevent reannealing, protect them from nuclease degradation, keep them in an elongated form, ready for replication. These proteins are non-enzymatic and act as DNA protectors. Summary table of Step 1. Initiation. Component function. Helicase breaks hydrogen bonds and unwinds the DNA helix. Replication fork Y-shaped structure formed during unwinding. Topoisomerase/gyrase relieves torsional stress and supercoiling. CBSEs stabilize single strands and prevent reannealing. Step 2. Priming. DNA polymerase cannot initiate DNA synthesis on its own. Primase enzyme synthesizes short RNA primers, 5 to 10 nucleotides long, complementary to the DNA strand. These primers provide a free 3-OH group for DNA polymerase to begin synthesis. Step 2. Priming. Detailed explanation. 1. Why priming is necessary. DNA polymerase, the main enzyme for DNA synthesis, cannot initiate the synthesis of a new strand on its own. It can only add nucleotides to a pre-existing 3-OH group. Therefore, before DNA polymerase can start working, a short segment of nucleic acid must be laid down to provide a starting point. 2. Role of primase enzyme. Primase is a special RNA polymerase. It synthesizes short RNA primers, about 5 to 10 nucleotides long, that are complementary to the DNA template strand. These primers are laid down in the 5'3" direction, because nucleic acid synthesis always occurs in this direction. 3. Function of the RNA primer. The RNA primer provides a free 3-OH group that is essential for DNA polymerase to start adding deoxyribonucleotides, dNTPs. Once the primer is in place, DNA polymerase can extend the chain by adding DNA nucleotides. For priming on both strands. On the leading strand, only one RNA primer is required at the origin of replication, because synthesis is continuous. On the lagging strand, multiple RNA primers are required, each for initiating the synthesis of individual Okazaki fragments, because synthesis is discontinuous. 5. Important characteristics of primers. Made of RNA, not DNA. Later removed and replaced with DNA. Essential for initiation of DNA replication, even though they are temporary. Enzyme summary for Step 2. Enzyme role. Primase synthesizes RNA primers, 5 to 10 nucleotides, complementary to DNA strand. RNA primer provides free 3-OH end for DNA polymerase to begin DNA synthesis. Step 3. Elongation. DNA polymerase 3 in prokaryotes, or DNA polymerase alpha, delta, and epsilon in eukaryotes. Add complementary nucleotides in 5'3" direction. Template is read in 3'5" direction. 2 types of strands form. Leading strand. Synthesized continuously toward replication fork. Lagging strand. Synthesized discontinuously away from the fork in short fragments called Okazaki fragments. Step 3. Elongation. Detailed explanation. 1. Enzymes involved. In prokaryotes, main enzyme is DNA polymerase 3, high processivity, fast synthesis. In eukaryotes, DNA polymerase alpha initiates DNA synthesis, adds few nucleotides after RNA primer. DNA polymerase delta elongates lagging strand. DNA polymerase epsilon elongates leading strand. 2. Direction of DNA synthesis. DNA polymerase adds nucleotides in the 5'3" direction. The template strand is read in the 3'5" direction. This polarity is critical for enzyme activity and strand formation. 3. Mechanism of nucleotide addition. DNA polymerase uses dNTPs, deoxyribonucleoside triphosphates, ATP, DGTP, DCTP, DTTP. These nucleotides pair with complementary bases on the template. Binds with T, G binds with C. A phosphodiester bond is formed between the 3-OH group of the growing strand and the 5' phosphate of the incoming nucleotide. The energy for this reaction comes from the cleavage of two phosphates, pyrophosphate, from the dNTP. 4. Types of strands synthesized. Leading strand. Synthesized continuously in the same direction as the replication fork movement. Requires only one primer at the origin. Carried out by DNA polymerase III, prokaryotes. DNA polymerase epsilon, eukaryotes. B. Lagging strand. Synthesized discontinuously, in the opposite direction of fork movement. Made in short segments called Okazaki fragments. Each fragment begins with a new RNA primer. Carried out by DNA polymerase III, prokaryotes. DNA polymerase delta, eukaryotes. 5. Okazaki fragments. Size of Okazaki fragments. 1,000, 2,000 nucleotides in prokaryotes. 100, 200 nucleotides in eukaryotes. Later joined by DNA ligase. 6. Proofreading ability optional add-on. Most DNA polymerases have 3'5" exonuclease activity to remove mismatched bases. Ensures accuracy and fidelity of DNA replication. Summary table. Elongation. Feature leading strand, lagging strand. Synthesis direction toward fork, away from fork. Type of synthesis continuous, discontinuous. Enzyme eukaryotes. DNA pol epsilon, DNA pol delta. Needs multiple primers. No, yes. Forms Okazaki fragments. No, yes. Step 4. Primer removal and filling gaps. In prokaryotes, DNA polymerase I removes RNA primers and replaces them with DNA. In eukaryotes, RNA primers are removed by RNase H and DNA polymerase delta fills the gaps. Step 4. Primer removal and gap filling. Detailed explanation. 1. Why primer removal is necessary. During replication, RNA primers are used to initiate DNA synthesis. These primers are composed of RNA nucleotides, not DNA. RNA primers must be removed and replaced with DNA nucleotides to ensure a complete and accurate DNA molecule. 2. Process in prokaryotes. A. Coli. Key enzyme. DNA polymerase I. DNA polymerase I has two functions. 5' to 3' exonuclease activity to remove RNA primers. 5' to 3' polymerase activity to fill the gap with DNA nucleotides. Mechanism. DNA pol I binds at the RNA-DNA junction. It removes each RNA nucleotide one at a time, moving 5' to 3'. Simultaneously, it adds corresponding DNA nucleotides in their place. 3. Process in eukaryotes. RNA primer removal. RNA primers are removed by RNase H. Specifically degrades the RNA strand of RNA-DNA hybrids. Flap endonuclease I. FN1 removes any flaps or extended RNA fragments. Gap filling. The gaps left after primer removal are filled by DNA polymerase delta using the template strand. 4. Result of primer removal and filling. The DNA strand becomes continuous, with all RNA regions replaced by proper DNA sequences. The sugar phosphate backbone, however, still has a Nic, unsealed phosphodiester bond, at the junction between replaced DNA and adjacent Okazaki fragment. This is resolved in the next step, step 5, joining by ligase. Enzyme summary table. Organism enzyme for primer removal, enzyme for gap filling. Prokaryotes DNA polymerase 1, 5' to 3' exonuclease. DNA polymerase 1. Eukaryotes RNase H, FN1 DNA polymerase delta. Step 5. Joining fragments. DNA ligase seals the nicks, gaps, between Okazaki fragments on the lagging strand by forming phosphodiester bonds. Step 5. Joining of fragments. Detailed explanation. 1. Background: why joining is required. After RNA primers are removed and replaced with DNA in step 4, the newly synthesized DNA still has discontinuous nicks, gaps, between segments. These gaps are especially present on the lagging strand, between Okazaki fragments. Although the nucleotide sequence is complete, the sugar phosphate backbone is not fully sealed. 2. Role of DNA ligase. Primary function. DNA ligase is the enzyme that joins the adjacent DNA fragments by forming phosphodiester bonds between the 3-OH group of one nucleotide, 5-phosphate group of the next. Mechanism. DNA ligase uses ATP in eukaryotes, or NAD in prokaryotes, as an energy source to catalyze the bonding. This creates a continuous DNA strand with no breaks in the sugar phosphate backbone. 3. Significance of ligase action. Completes the replication process by sealing Okazaki fragments on the lagging strand, repairing any nicks in the leading strand if present, ensures DNA molecule is structurally intact, stable for packaging into chromosomes, ready for transcription and cell division. Enzyme summary. DNA ligase. Enzyme function, energy source. DNA ligase forms phosphodiester bond to seal nicks in DNA backbone ATP, eukaryotes, NAD prokaryotes. Key terms to remember. Nick. A break in the sugar phosphate backbone between adjacent nucleotides. Phosphodiester bond. Covalent bond connecting the 3-OH and 5-phosphate groups of neighboring nucleotides. 5. Key enzymes and proteins in DNA replication. Enzyme/protein function. Helicase unwinds DNA double helix. Topoisomerase, gyrase. Relieves supercoiling ahead of replication fork. SB proteins stabilize separated single strands. Primase synthesizes RNA primers. DNA polymerase 3, prokaryotes. Main enzyme for adding nucleotides. DNA polymerase I, prokaryotes. Removes RNA primers and fills the gap. DNA polymerase alpha, delta, epsilon, eukaryotes. Main enzymes for eukaryotic DNA synthesis. RNase H, eukaryotes. Removes RNA primers. DNA ligase joins Okazaki fragments. 6. Replication fork and bidirectional replication. Replication is bidirectional. Two replication forks move in opposite directions from the origin. 7. Okazaki fragments. Short DNA fragments, 1000 to 2000 BP in prokaryotes, 100-200 BP in eukaryotes, synthesized on the lagging strand. Named after Reiji Okazaki. 8. Proofreading and fidelity. DNA polymerase has 3' to 5' exonuclease activity for proofreading. This ensures high fidelity, accuracy, of replication. Mutation rate is about 1 error in 10 nucleotides. 9. Energy requirement. DNA replication is an energy-consuming process. Energy is supplied by dNTPs, deoxynucleoside triphosphates, like DATP, DTTP, DCTP, DGTP. Breaking of high-energy phosphate bonds releases energy to form new bonds in DNA. 10. Direction of replication. Feature direction. Template reading 3' to 5'. New strand synthesis 5' to 3'. 11. Result of DNA replication. Two identical double-stranded DNA molecules. each with one parental old strand and one new strand. Enables accurate transmission of genetic material to daughter cells. Bonus facts. In prokaryotes like E. coli, replication starts at a single orisite. In eukaryotes, multiple orisites exist due to large genome size. Speed. E. coli. 1000 nucleotides/SEC. Human cells. 50 nucleotides/SEC.